स्टूडेंट आस्क सर आपने मेरा ये किया नहीं है, ऐसे पार्किंसंस हो रहा है, any photograph, and that's a problem of dopamine. You know, you get addicted with this, and that is usually come. Good to see all of you in the morning. Here, मैडम जी इधर आ जाइए. है कोई उधर? You can keep these bags here in the side. Come to the iron. Parkinson's disease is usually a problem of older ages. It starts around 50, or even sometimes nowadays you can see in very early days. Okay, I'll tell why. But alarmingly, this is the now highest rate of neurodegenerative diseases. Earlier, it was like Alzheimer's disease. Okay, now Parkinson's disease is catching up. Okay, that way, because it comes mostly from the toxins we are inhaling, we are using. If you just buy a spinach from market, you don't know how much herbicides or pesticides are there, and that has a direct impact on Parkinson's disease. It's mostly a sporadic disease. What is sporadic disease? Any idea? नहीं है. It may happens. Okay, in a particular, say time to everybody's life, and it may not be. There are only few. There is a gene called Parkin. There are very small percentage of. I think. Can you come here? You are fine. Okay, ठीक है. So there are very like 10% or less that is genetic. Otherwise, it's sporadic. In a family, suddenly, in a generation, it may. आप वो थोड़ा बंद कर दीजिए दरवाजा. हाँ, तो it may happen. So that means why it is happening? It is happening because you were exposed to something in your lifetime. Okay, and this has now been identified about. I'll give some data with the pesticides, herbicides, and many other things. Even recently, it has been found the kind of pollution we are going through. Okay, that is causing a lot of trouble also. So we'll come back. Now, what is the connection of iron? I'll come back. Why I am talking about iron? Because 100% of this Parkinson's disease are linked with iron accumulation in the brain. Okay, and somehow it is less. If you can take that iron out, the Parkinsonism would not be better, but it will be static. Why? Because your brain cells are being already damaged. That cannot be again regenerated. So that's why these are known as neurodegenerative diseases. Okay, so what we'll be talking about today is a bit of iron and how it can be accumulated in the brain. Okay, so if that is one of the causes of Parkinsonism, and if that can be controlled in the future and from the early days, so it would be less or it will not cross the threshold. That's the idea. There is no therapy still so far. So if you know that, and that's why one of our goals of this, our lab, is to understand the role of iron. Now, if I ask you what is iron? Iron. And okay, and what is its role? Is it important at all?
It carries oxygen to all our tissues and throughout.
Anything else?
Oxidative stress.
That can increase oxidative or participants. Anything else? Anemia?
Yes, sir.
And how many of you are anemic? Any idea? I'm telling you, in India, 65% women are anemic. It's a huge problem. And not necessarily this is due to iron deficiency. I could have talked about that part, but I think this is also interesting. I'll tell you what I do in the lab. So iron deficiency also may happen. I say about how much we have population now, whole world, 9 billion. If it is 1/4 is suffering in iron deficiency. Okay, and out of that, half, about 50%, is due to they cannot take iron. So 1 billion people are suffering not due to iron deficiency, but there is still anemia. That happens due to infection, inflammation, and I am not going to talk about it today. So let me see why iron is a very interesting molecule, because it is redox active, because it can go ferrous and ferric. Okay, that's its strength and weakness. So if you say that way, that this is, have you anybody heard, have you read that A Tale of Two Cities by Charles Dickens? Have you heard the name of Charles Dickens? Huh? Who was he? Parkinsonism आपको हो गया. Okay. So Charles Dickens was a very, very famous writer. Okay, he also wrote David Copperfield. Some of you may read it. So Charles Dickens wrote one of the most famous novel. You should read that. It's a tale of two cities, the best part of a city and the worst part of a city. Okay, and he started the novel with, this is the best of the time, this is the worst of the time, and that any time you can see this is happening. So that's why I say iron is the best of the metal and worst of the metal. Okay, so this is anyway is the fourth most abundant metal, and it is needed by all the organisms for electron transfer and energy generation. So if there is low iron, nobody can survive. Nobody. Without oxygen, there are many organisms can survive, but without iron, nobody can survive. And why it is worst of the metals? You know, this is highly insoluble. That's why you get rusting. Okay, insoluble means 10 to the power minus 17 molar solubility product in aqueous pH. Means around, if you put into the water, it never resolves, it never dissolves, it precipitates. Okay, so it is fourth most abundant element, but still 1/4 of the whole population are suffering in anemia. Okay, if it is this much, so there should not be any anemia. It's feeling like you are during a flood, you are in a boat, but you cannot drink. There's all around, you cannot drink, it is contaminated. Similar with all around there are iron, but you cannot utilize. Main problem is it is insoluble. Okay, I'm not going to that chemistry right now, but it can participate because of this redox. It can participate in reactive oxygen species generation and can damage. And one of the most active area of research in everywhere is ferroptosis, iron-mediated cellular death process. This is the only mechanism by which cancer stem cell can be died. Okay? Because all the cancer comes back because it comes from the stem cell. So if you can really kill cancer stem cell, then you have another answer for cancer. And that's people are so much excited and working on. Okay? We are fortunate that we are working on this area for quite long. So just to remind you, all of you know this is 26th element in the periodic table. Chemical symbol is Fe, molecular weight is around 56, and it can oxidation be -2 to +6. I am not going very details of the Werner chemistry, and very useful for redox chemistry in the body. Any electron transport happens. It's like electricity. If there is switch off, we know electricity. That means electron transfer is affected. Okay. Similarly, if there is no iron, this electron transport will be affected. So even you have highest amount of glucose, you may not get any ATP generation. Okay, so that is the important. Forget about oxidation. Even a bacteria, if they don't need any oxygen, but they have to depend on iron. So one of their ways is to, when they infect, first thing what they do, they snatch iron from your host cell. That is another mechanism. I'll not talk much about it today. So to tell you about iron, there are three important mechanisms for an iron balance. So iron balance is important because if it is less, you don't get ATP, you don't get oxygen transport. If it is more, it can cause oxidative damage and may cause to Parkinson's or that kind of diseases. Almost every disease is linked with the iron. And you will learn today is something new, it's called iron accumulation. Usually we are very much aware about iron deficiency. Okay, but iron accumulation, even in diabetes, if you check your liver with MRI, there is more iron in your liver. So diabetes has very old time, from very old time there is a solution for diabetes. If you let your blood out, it's called phlebotomy. So if you let your blood out, then you get insulin sensitivity. Diabetes will be coming down. It will be reduced. Okay? But still don't know, mechanism is not known, but it will come back again. So it is good to sometimes give your blood, donate your blood. That will help to control your diabetes also. Okay. So if in a cell, what our hypothesis is that iron is accumulated, and this is contributing to Parkinson's disease. Not only means who are people are working in this field. So if we want to know how iron accumulates, we have to think about three mechanisms. One is uptake, which is coming in. One is storage. If the store is affected, then that can also contribute. And another is release. So in any place, say if you have a tank where the water is coming in, how much water will be there will depend on what? How much it is coming in, how much storage it is, and how much it can go out. Similarly, in any cell, you think about is in a water tank. Iron is coming out through a mechanism called transferrin receptor. Okay? Transferrin receptor 1. Today we will not talk much about transferrin receptor. Okay? We'll talk more about ferritin, which is iron storage, and ferroportin, which is iron release. Say, if somebody has lot of money coming, or even whatever, but if don't have a place to store, what it will happen? That's a problem. Okay, you cannot withhold. So that means that iron cage is important. Or how much, if you cannot, say your father, you are here in Delhi, and your father or mother send you good amount of money, but telling that you cannot spend any single penny, what it will happen? It will scratch you. And you will feel really bad. So that means ferroportin, if it does not work, and just to know whole mammalian system and eukaryotic system, there is only one mechanism by which a cell can release iron, that is ferroportin. That's why it is very, very important. Okay? So in between what is happened, to let you know, when iron comes in, for around 2 hours, cell keeps it in a kind of a transition level. It can't decide what it will do. It has to decide. So that is called labile iron pool. Okay? And then if it is extra, it goes for iron storage. It is extra, it's for release. Otherwise, oxidative damage will happen. And in between, iron goes for heme synthesis, these and that, etc. And ferritin also holding iron. Say when you are not taking any food, you need iron every moment. You are like billion heme number is being synthesized. Where from iron is coming? It is being degraded by ferritin, because that's where. Say if you are not having any income, where from you will spend money? From your storage. So that's why it is important to store iron and store money, both. Okay? Although money is very abhimani, any point it can go out. Similarly, okay. So this you more or less now know. Now what it is happen if there is an extra iron? So in presence of, this is a very, very famous reaction called Haber-Ruiz reaction. It is first time somebody told in regards to industrial chemistry. First it was discovered in 1892. Then again it has been re-emphasized in 1908. Okay, and after 100 years, it's still people don't know everything about it. It's very only thing it's known that when there is iron and superoxide, even the superoxide radical, then it can make hydrogen peroxide, and finally it makes an hydroxyl radical. And this hydroxyl radical is very dangerous because we have no defense against hydroxyl radical. You have defense against superoxide radical, you have SOD, and then for H2O2 you have catalase peroxidase. For iron you have ferritin and all these carrying protein, which is, it's like a cradle, they will keep you. But for hydroxyl radical, it is like a tokamak which will go and hit the ship. Once it is switched off, all these ballistic missiles. So it's similar, like if hydroxyl radical is formed, it has to degrade somebody. Okay, and mostly these cause lipid peroxidation, that means your membrane will be bursted. It can cause protein oxidation, that means it is impacting on aging. Oxidative DNA damage, this is one of the great mechanisms which is known so far for generating cancer. Okay, and in whole it can cause cellular or neuronal death. It's known as ferroptosis pathway. This ferroptosis pathway, the name has been only given in 2012, just about 13-14 years before. So I'm not going much to talk about ferroptosis, just to tell you the diseases. This is a list of diseases involving iron. So first I keep anemia because 2 billion people are suffering at any moment, okay, and related disorders, and hemochromatosis and cancer. This is tissue iron overload. So one way there is low iron, the other way you have more iron. It is very well known the cancer patients have more iron storage. So there were some experiments were done in Africa, the Bantu tribe. They usually take the alcohol from iron pot. And it has been found they are highly susceptible to malaria because of high iron, they are infected, and they have 400% chance of eye cancer. Okay, so some of them are kept or given. This is their culture, so they don't want to take. But still, for some people have given some kind of rewards and say don't take. Okay, and say that amount of population, the cancer chance is far less now. Okay, so then you can see this comes from the iron overload. And diabetes type 2, obesity and fatty liver. Now there is a huge disease now, 50-60% people are anywhere in the whole world suffering, called non-alcoholic fatty liver disease or steatosis, steatohepatitis. Maybe you have heard a bit. This is kind of a liver inflammation where iron level is also high. So that contributes there. And all neurodegenerative diseases, Parkinson's, Alzheimer, prion, dementia, it's all related to iron related. Cardiovascular diseases, okay, and infection, all infection including virus, I didn't put it there, bacterial and parasite, and inflammation. Okay, there is a direct link. There is one of the neurodegenerative inflammatory disease called multiple sclerosis. How many of you heard of multiple sclerosis? Anybody else? Yes, good. So what is happened, they have found wherever the multiple sclerosis in the brain, there is iron foci. Okay, so that means it is directly contributing to the multiple sclerosis. So this is a therapy now, that how you can reduce the iron from the brain. But once it's deposited, it's very difficult to take it out until it damages everything. So we have to know in normal condition how iron can be accumulated. Okay, and just why some are yellow and some are white, because these white areas I do research in my lab. So I work on iron inflammation, particularly brain inflammation related to multiple sclerosis, parasitic infection and leishmania and related diseases, Parkinsonism, diabetes type 2, obesity and fatty liver, and hemochromatosis and cancer. Again, brain cancer, where we are trying to understand. Okay. So I unfortunately I have a lot of interest, because it's common. So you can do many things. So these are, now we focus on brain. So brain iron deposition and disease. So Parkinson's I mentioned, Alzheimer's similar way, multiple sclerosis, neurodegeneration with brain iron accumulation. This is another disease. Okay. prion disease, anybody knows? Mad cow disease or prion disease? You have heard this, okay? So that is also related to, this, what?
Accumulation of iron.
Iron, yes. So aceruloplasminemia, this is, I will come about a bit. This is a copper containing protein. So copper and iron are cousins through the evolution, okay? So if there is no copper, then there will be anemia also. Again, that is another. And dementia, you are forgetful. This, when people are getting aged, they are more forgetful. One of these problem is iron accumulation. And why? Let me tell you. Because how your neuron fires. So you want to recall something. There is a place called tripartite junctions. Any idea in brain what is called tripartite junction? Where blood, astrocyte, brain cells and oligodendrocytes are together. And it has to, so where is the memory? Usually it's related to myelination of oligodendrocyte. And that myelination is also dependent on iron. But it has to fire. Say, to switch on, what you do? Switch on. So that firing comes from the ATP release of astrocytes. So if there is iron accumulated, that means that iron is not available for mitochondrial electron transport chain. So your ATP release will be highly affected. So that means brain will not fire. You cannot recall. You are trying. Lag raha hai, soch raha hai, nahi ho raha hai, because ATP is not firing at the right time. You take a rest. You can do a simple experiment. Don't take a sleep for 18 hours and try to remember and see what happens. You will see you can't remember. Okay, then you have a good 8-10 hour sleep and see immediately what is the name of daughter of Abhishek Bachchan, you can still remember that. You can remember every nitty-gritty, small, whatever possible, you can remember and see what happens. Because now, because of rest, your metabolism is slowed down, ATP is accumulated, okay? And there are more mechanisms. So this is one of the mechanisms. It is, see, in biology there is not only one hypothesis, okay? And it's everybody, everything is so much related. Okay. So now let's come a bit on Parkinson's disease. So this is just a very quick one. So Parkinson's disease belongs to a group of conditions called movement disorders. So people cannot grab things, they cannot move, okay? And it is both chronic and progressive. And first described in 1817 by Dr. James Parkinson, that's where the name came is, the Parkinson's disease. The disease affects approximately 2 of every 1,000 people and most often develops after age 50. We are not there, fortunately. It is good to remain young. You will not be, Parkinsonism will not much affect you. Sometimes Parkinson's disease occurs in younger adults and is rarely seen in children, but it affects both men and women equally, so it is completely gender equalized. So there is not much problem, equally distributed geographically and between socio-economic classes, although this is not really correct. Okay? If you go and Google it, and you will find there is a Parkinsonism belt in US. Whatever the country US is, it is very frank. It talks about its negativeness. In India, everything we hide under the carpet. Okay, so you see there is a belt from Texas. It is going to the Michigan this way. That is highly Parkinsonism zone. And why? Because that's like Texas itself. It's highly dependent on herbicides and pesticides and using chemicals. Okay, so there is a link there. So that means in that case, even people who are younger, if they take too much of these pollutants, too much of these chemicals, they may have a chance of inducing Parkinsonism. So what are the major symptoms, sir? You know, so I don't have to say this is not a physiology class. So there are obviously movement disorder, forgetfulness, and people can't even move. They cannot move even to here to here. Okay, and so where it is happened, what is happening? Somebody told here from that region that it is a problem of dopamine. So what is that? So dopamine is a fantastic hormone. If you are having fun, your dopamine is more. Okay, and if you are not enjoying my class, your dopamine will be that way. Okay, it will be so jayega. So you cannot move, having more sleep, etc., etc. Okay. Now see, is dopamine synthesized in hypothalamus in the region substantia nigra, just here. Okay, and they found this is about only 20,000 this kind of neuron. And if these 20,000 or even 50% are affected, then we are in trouble. We cannot recall things. We cannot. This movement is particularly affected. So this substantia nigra that produce chemical. So what is happening in Parkinson? Parkinsonism is going up, and dopamine is going down. Now if we hypothesize, if we have more iron deposits there, so that iron is causing oxidative stress to kill those neurons. Okay, and that's why dopamine is not doing well. So nowadays there are therapy called L-dopa, and there are the different kind of carbidopa are available, but those dopa are not so great. Everything has, then life would have been so nice. So our job as a scientist, what we try to understand, why this has happened. If you know 5 reasons, and if you take care of that, anyway, the disease will be at least stopped or come later. That's why we want to know that why these cells are being died. So there are 2 major things, if I do remember correctly, because I'm susceptible to Parkinson's. So 2 things happen. One, mitochondria does not function well. Okay? And iron accumulation is there. So today we'll try to talk about at least 2 important cells in the brain. One is astrocytes, will come there, and second is neuron. Which case, how dopamine controls iron to run everything smoothly. And when there is no dopamine, whether that can cause a problem, that is the goal. So dopamine constitutes about 80% of brain catecholamines. So there are, how many of you know about catecholamines? What are those? In brain? A neurotransmitter, yes. What are those? Any name? One is in front of you, dopamine. So you should start with dopamine. Serotonin. Okay, good. So you know. And this catecholamine synthesis also depends on iron. So if there is no iron, catecholamine synthesis will be highly affected. So you see how many dangers are there. Okay, there are many dangers. So if there is no iron, so what is accumulation? Like you have 100 things, but you cannot really get hold of them. So somewhere it is not available. You are taking it, it's not so. Anyway, it is not going to a pool which is not usable. So this is one, that means your, again, dopamine synthesis will be down. Not only the neuron, so when it starts not with the neuronal damage. It starts with the iron is not available. Somewhere it holds. So we'll come there. Where? Okay. So DA synthesis is affected in neurons in PD. Patients and model animals are detected with iron deposition and mitochondrial dysfunction. So we'll try to understand in 2 cells how it happens. These observations suggest an intimate relation between the DA and iron homeostasis. So that means iron is synthesizing DA, but whether DA is also doing something on iron homeostasis, okay, that was not known. So we have in our lab really focused on that area. So the hypothesis was there for long that one of the mechanisms to develop PD is iron-mediated oxidative stress for the damaging of neurons. But initial stage, was not described there. And mitochondrial dysfunction and subsequent cell death. This published in Science in 2003. Mainly environmental factors than genetic factor are found responsible for PD. I mentioned already, scientists linked exposure of toxins, particularly herbicides and pesticides, in developing PD. So if you go to PubMed, you know what is PubMed? You can search. So if you search, yesterday I searched pesticides and Parkinson's in PubMed, it's showing 2028 publications. So it's a huge amount. People are, and herbicides and Parkinson is 716. So that means there's a huge amount of evidence to show that Parkinson's is related to pesticides and herbicides exposure. And if you take diet containing higher amount of iron, then you have also likely to develop PD by 1.7 times. So if you see in doctors, they recommend that in late stage of life, you should not take a lot of goat meat where the heme is very high. So one of the reasons they say only about the metabolic disorder, but another way it is. People who are taking a lot of iron, they have a more chance of earlier aging and Parkinson's disease. So increased iron content in brain with aging. So if you just do MRI of the brain, your age and my age, you will see in my age there are at least 2 times more iron is accumulated here. Okay, so these are one, and iron and aging is another huge area. You know, people are lot of the time raw iron and aging, people are working on it. And this is a simple experiment. I'll not talk about details, but what they have done in a monkey, they injected MPTP. MPTP is a pesticide. Okay, simple pesticide they injected here and checked with the iron staining and see just by a single injection in the brain of MPTP, and MPTP is now supposed to be banned, but you can still get it is many ways. And in the other part, not injected, you see the iron staining is far less. So it is a direct experiment to show that iron can cause a problem. And MPTP is a standard way to make Parkinson's disease in animal. If you inject MPTP, after 2 months you see people are not moving, they are doing this limbing, and that dopamine is here. So this is a direct way that people have found. I'm not explaining the next one, it's good. So now let's get back to the brain. What is the architecture of the brain? Architecture in sense of only the brain cells, not the skull and everything. So usually the cells, so we are linked to the neuron through astrocytes. There are glial and neuronal cells. How many glial cells are there? How many types? Astrocytes, microglia, and oligodendrocytes. Okay, so these astrocytes are like border security force. Okay, whenever things are happening, coming in, coming out, they have to check. So through the blood-brain barrier, when things are going there, including iron, they have to first go through the astrocyte. And astrocyte, there are another term I have to tell you, is that what are the neurotransmitters? Dopamine, norepinephrine, serotonin. Why they are called neurotransmitter? No, these are being synthesized and released from the neurons. That's why neurotransmitter. Similarly, so how they crosstalk? They do Toretokka cells. You send something, I'll send something. So astrocyte sends ATP and a lot of things. For today, we'll talk about ATP only. So this is called gliotransmitter. This is a new term maybe. So neurotransmitter comes from the neurons, and glia, which is going out, that's called gliotransmitters. And ATP, like astrocyte, is a very unique cell which can secrete ATP. Most of the cells are ATP is synthesized, and if it is extra, then it is kept as fat for future But here, this is a cell, there is not much, it just releases ATP. And that's why your neuron fires. Okay. So, these astrocytes are linked with neurons, and these are the blood vessels. So, if I have to take iron and it has to go to the brain, from the blood vessel it will come to the astrocyte, and then astrocyte to neuron, because astrocyte will check it, and accordingly they will release what happened? No ATP. Tell. And in between, hopefully the ATP comes back here. Chalo, thank you so much. Okay, so this relation is clear to you. So let's go to the next one. So now you think this is the blood vessel. This blood vessel has to send things to the neuron, presynaptic, and these are tripartite junction. Okay, where glutamate also has a big role to play. So this will only go through the in-between border security force. It's called astrocyte. So if iron has to go, or iron has to come out, both ways, the astrocyte will make the checking and everything. Are you taking more iron? Are you taking less iron? What is exactly needed? That they will help. And in astrocyte, now if you see there is iron uptake, as I mentioned, iron storage and iron release. So if from brain iron is coming, here it is a new protein called DMT1, which is a cousin of transferrin receptor. Okay, that's very specific for astrocyte because it's a specialized cell types. And then iron comes in, first what I mentioned is labile iron pool, and either they will go for heme synthesis, iron sulfur cluster complex generation, because that's important for many things. Okay, and we'll talk about heme synthesis. Iron sulfur cluster will not talk much today. The time is limited, and the rest will go to ferritin. And for release, the protein is, which is I say gate, through which iron is released, but gate is not digital. Okay, so there is somebody else which is a gatekeeper here in astrocyte called ceruloplasmin. Do you remember aceruloplasminemia, where I mentioned about? So when there is a gene mutation in ceruloplasmin, this aceruloplasmin, there is no ceruloplasmin. So iron remains here, it does not go to neuron. And that causes a lot of problems. Okay. Fortunately, India, this genetic disease is not much. So this is for the iron release. Now, we'll come back to ferritin. What we have found that dopamine has no role in regulating iron uptake. So if there is no dopamine or dopamine, same amount of iron will come in. Now what happens to ferritin? Now ferritin is a very, very interesting molecule. Just to tell you, I'll take probably a little bit more time because I talk a lot of faltu things. Okay. So ferritin, last week there is a paper came out in Science. Okay, I'll bring all these issues, which says that how a pigeon or bird can locate to go back. If you throw them somewhere, they will come back. There is a bird migration. So in long time, people were working on it, and then science, this has been shown that in liver there is a cell called macrophages. You know Kupffer cells in brain. So how much ferritin is there? Ferritin is iron storage. So it stores a lot of iron. That means it has a relation with magnetism. So they found if they have lower ferritin, they cannot go back from where they are come out. So what they are doing with that amount of iron, they are cross-talking with the geomagnetism. This is one part. Ferritin is so interesting. When COVID, you have a cytokine storm. You don't have, fortunately. Some people have cytokine storm and died. So that morbidity it was clearly linked with the ferritin level in the blood. So if you check ferritin level is higher, it is still good for any inflammatory diseases. So that's a good biomarker. Okay. If somebody has more ferritin, more iron than 4,500 iron atoms, they should go and check doctor for whether they have cancer or not. Last year, 3 papers came and same issue of Nature, which shows the amount of iron and the ferritin determines, just believe, determines the sex. So during pregnancy, how much iron is going? I don't know whether India will start this working or not, but they say the amount of iron determines whether it will be male or female. Okay, so these are, you can see how much exciting the field is. So this is very, very important, and we'll talk about ferritin today a bit. So this one molecule may store up to 4,500 iron atom for 2 reasons. When you have a demand, you can release. It can degrade it. This is called ferritinophagy. Ferritin goes to autophagic degradation at lysosome. It's a very famous term called ferritinophagy. Okay, and that iron now can go for. So if you don't take iron for some time, your body is good enough to still make heme and iron sulfur cluster and many things. Okay. And this is regulated by a translation mechanism in response to higher iron. So when there is more iron, in 5' UTR of this gene, there is an iron sensing element. Where iron sensor, because more iron, so these will come and sit down or go away. What is happened? And accordingly, there will be more translation. Why translation? Why not transcription? Because if you transcribe something and then send for translation, you need a lot more time, because that long time iron can make damage. So it has to be in situ then and there. Okay, so that's why iron, and this is the first discovery of mammalian iron translation mechanism. So what you read in textbooks, it mainly came from ferritin and transferrin receptor, mainly ferritin translation receptor. So, but when it is extra, when for whatever reason ferritin is degraded, you are under inflammation, ferritin is degraded, that extra iron can cause ferroptosis and cause cell death. Okay, so ferritin H is again a marker of ferroptosis pathway. Ferritin should go, if it goes down, means it cannot store. So that's why it is very, very one of the major mechanisms that controls cellular iron availability and cell death. So if your ferritin is doing well, cell is doing fine. If it is not doing well, cell will die. Okay, so this is a death called ferroptotic death. So earlier people thought, so what we wanted to do? We want to check what happens to ferritin if we treat astroglial cells by dopamine. You know what is Western blot analysis? How many of you know? No Western blot? Pata hai. Good. So anyway, so Western blot is the amount of protein you will know by using antibody. It's a blot analysis. So we checked by Western blot. We were thinking there will be upregulation. So we took 3 different cells. This is U87MG, is an astroglial cell, and you see with increasing doses of DA, by any chance DA can be, DA can be go up to 200 to 300 micromolar when there is a surge. So we are pretty inside the physiological concentration. Okay, and you see ferritin H. So this has 2 subunits, ferritin H and L. Okay, both are going down, and this is a positive control. What I said, when there is an extra iron, what it will go? Ferritin will translationally go up. So there is more ferritin. So we added 1 iron salt, it called ferric ammonium citrate, and you see both are increased. Similarly, we found with rat cortical astrocytes. So we isolated rat cortical astrocyte and treated with them. And finally, what we did, we took human iPSC. Okay, the pluripotent stem cells, because whatever you can do, you can make into astrocyte, you can make into, because there are Yamanaka factors are there through which you can do. So we have done this, and there also we have seen ferritin H. I am not showing ferritin L data, so it is going down. So what we found, that we add dopamine, the iron storage is going down. Okay, so we wanted to know the mechanism. So we checked RNA level, there is no change of RNA level. So if your mRNA are same, then either protein is degraded or the translation is blocked. Got it? Clear? So then we check the protein degradation assay by cycloheximide chase assay. What is cycloheximide chase assay? Again, if you go back to your water storage system. So water is coming in and water is going out. So if you stop coming in, and how long it will take to go 50% down, you have different kind of, that tub or whatever, and you are doing experiment on that way. So if you do, then how much is the 50%? That is called half-life. So how do you stop transcription or translations? Here you have to stop translation as uptake. We added cycloheximide, which is an antibiotic to stop ribosomal assembly. So translation will not happen. And then we chase, usually ferritin is very much stable. We chased up to 9 hours and see there is almost no change in the protein level. But when you added dopamine, you see within 3 hours, more than half is gone. So it is telling that ferritin is being degraded. This is another assay, pulse chase. I will not tell you about it because of time. And then we check now whether protein is degraded by 2 different mechanisms. Can you tell what are the main way of protein degradation?
Proteasomal ubiquitination.
Fantastic. Yes, proteasomal by ubiquitination degradation and in lysosome. Okay, so we checked if we block with proteasomal blocker, what happens? So if proteasomal mechanism is dependent, if you block the proteasomal activity, then ferritin would not degrade. Is it clear? So we added epoximycin, which is known to block proteasomal activity, and still ferritin is being degraded. So that means it must be through lysosomal pathway. So we added then many of lysosomal blockers. We just show one which is named as chloroquine. Do you remember anything about chloroquine? COVID se pehle. Good. How it does? Same mechanism. Yes, this is the same mechanism by blocking lysosome. Okay, because then heme is not degraded and Plasmodium cannot get the iron, because if heme is more degraded or ferritin is degraded, malaria utilize that iron for their growth. So this is the mechanism. Even artemisinin does the same thing. They are iron chelator. Now you can understand a bit more about the importance of iron everywhere, you can see. So what we got now, that it is due to lysosomal protein degradation. Now lysosomal protein degradation happens by 2 different mechanisms. What is called macroautophagy and another is called chaperone mediated autophagy. So anybody? No. It's difficult to get macroautophagy versus chaperone mediated autophagy. So macroautophagy means that there are a group of, let me tell you. So far what was described in the whole literature of the world, the ferritin is shown either by proteasomal mechanism or by macroautophagy mechanism. Okay, so we wanted to check what is new. So this is usually macroautophagy, means a protein has to be anything. It may be ferritin or whatever. What is happen? They actually starts with in a different whatever process. I will not go. When ATP is down, your AMP comes more, and that AMP is sensed, and that's why all these starts, because iron is again needed for the ATP generation. Okay, so that is essential. And so there is something, there are many family members there which contribute. So a lot of this protein contributes for the autophagosome formation. where the lysosomal degradation happens. So we took 3 of the members, one from this pathway, Becklin, then ATG7, ATG5. And see, when they have to, somebody has to work more, so their activity or number should be more. Okay? So if we do again by Western blot, if you see, numbers are not more. ATG5, ATG7, Becklin 1, not more. But as a scientist, you will always argue against you. If anything is wrong there, maybe their activity somehow. So what we did, we knocked down ATG5, ATG7. You know knockdown experiments? Okay, by siRNA knockdown. So you can see here that ATG5 and knockdown is far less, ATG7 knockdown is far less, and still ferritin degradation is still happening. So by 2 ways we show that it is not by macroautophagy. Why we have done 2 ways? Because whole world knows that ferritin is degraded either by proteasomal pathway. He has not slept well last night. Okay, so and you can go and take a bit more oxygen and come back. It's okay. You have an oxygen debt now. So don't mind. No, just sometimes you have to talk, otherwise it becomes very monotonous. So what we are against the whole world. They say, okay, this is the way to go Taj Mahal. I say, no, you can go to Bengal and come back to Taj Mahal. That is also way. But why? Question is why and how. So then we checked. Let us talk about what is called chaperone mediated autophagy. What are chaperones?
Proteins which are involved in the protein folding and maintaining the structure.
So that will give you an identity. Folding means it's a particular identity. Chaperone will say, like when they came, they basically chaperoned me to do this work. So they carry and gave me, okay, these are the jobs you have to do. They opened the slides, they did, etc. This is a role of chaperone. So this is chaperone mediated autophagy, means there are particular 1 or 2 proteins. In this case particularly, it's called heat shock chaperone HSC70, which find that they are always scanning in which of cytosolic. So this happens mainly for cytosolic protein. Okay, so which cytosolic protein has a sequence like KFERQ, a sequence motif. Okay, KFERQ. So this, if they find, if it is more, then they bind it. Just grab like this, okay, and take in front of lysosome. And lysosome, there are different proteins called LAMPs, LAMP1, LAMP2, LAMP3, etc. What are these? These are lysosomal associated membrane protein. So what they do, they are like standing like this. They dijiye. So as soon as HSC brought that criminal, just grab and took it within the lysosome. And lysosome, there is an acidic atmosphere. Okay, so that means if we have to check that whether ferritin is going through CMA pathway, you have to check whether it is binding with HSC, because that has to grab. Then whether it is binding with the LAMP, particularly LAMP2A to B to C. Okay, and that's why. And if you block that binding, then the degradation will not happen. That's the proof. So first what we did, we checked whether LAMP1, that lysosomal associated proteins are increased by dopamine or not. So the platform, if a ship is coming, or if a plane is coming, that platform should be ready. Okay, so to degrade ferritin, the platform is LAMP. They should be taken, and these, you see, they are increased by dopamine. So ferritin is down and LAMPs are more, whereas macroautophagy markers are not changed. Okay, now what we did, we knocked down LAMP2 and LAMP2A, and you see now ferritin degradation is blocked. So that means this ferritin is happening through the lysosomal pathway. Is it correct? Tentatively correct. What is the main things are left out? Are they binding? It may happen separately. Science is you have to really prove and prove and prove. Okay? So if you see, it is called immunoprecipitation experiment. If they are together. So if you pull one, other will come. I am from Bengal. Last 5 years I am not there. But there is a labaj there, proverb, that if you pull your ear, the head will also come. That's simply immunoprecipitation. Okay. So if you pull one of them, all our member all together will come. Say like this is the way criminal investigation happens. You have to find somebody. So there is a group of criminals. You just find one. All will come slowly. So similarly, so what we have done, we immunoprecipitated with LAM2 antibody because this is that platform. We have done with everything and just showing one. So when we pull down with LAM2, you see it's still binding with ferritin H, binding it itself, obviously, LAM2 and the chaperone. So all three critical components are part and they can be part of this when there is more dopamine. Okay? And you see input, how much? You can, when you are loading it, there may be some changing. So you have to show by input how much you have actually done. And you can still see the ferritin H is down. Input may come hai, but binding almost same. Okay? So next, again, science is very difficult business, but it's a lot of fun. It's like a detective within the cell. That's the fun I see. If you are fond of, detective stories, you should be a scientist. Because what's happening? What is the next? How to prove better? So, what I mentioned that this chaperone-mediated autophagy happens when there is KFERQ-like sequence. So, is there any KFERQ-like sequence? So, what is KFERQ-like? Signature motif. So it is, they say, after a lot of work, so there should be at least 2 positively charged residues. So you see K and R in that sequence. There should be, at least 2, maybe up to 2 of hydrophobic residues, like F, and there should be a single negatively charged residue, E, glutamic acid, and Q at N or C terminus. Here it is C terminus. So we checked, there was no classical KFERQ in ferritin. But then with this kind of motif, we checked in ferritin and found that I think there are some changes happen, but changes means some distortion happen with this and the particular this one. And then we found actually moved QDIKK. This sequence is present in human, rat, mouse, bovine, and even plant, maize. So that means it evolutionary concept, and it may go to chaperone-mediated autophagy. And this is exactly matching the way it is. I'm not explaining because of time, you just believe me, okay? Now, again to prove it's there to kya hua. So what we have done now, if we disturb that sequence, is it binding to HSC70? That's the way. So what we have made, we clone ferritin H with a tag called flag. Okay, you know the tagged system, like histidine tag, flag tag, biotin tag. Okay, so we clone the ferritin H and put into a flag vector. So now this ferritin H lost its bit of identity, but it can, with the flag antibody, you can pull it down. So now we pull down, so we made 2 ferritin, one is wild type and the mutated one is from QDIKK. These are the 2, Q should be there, must if you see from there, and one is the single negatively charged residue. So D is also negatively charged residue, so we changed into this AA. Now see this binding with HSC is completely lost. So this is kind of we have added a lot more proof, but I am not telling. This is to tell you, convince you that this is happening because we are the first one in the whole world. Ferritin is a very highly research topic, but we are still the first one in the whole world to show that ferritin can go through chaperone-mediated autophagy. Okay? So that's why we have done a lot of these experiments. Okay, now ferritin is inside the lysosome. Iron has to come out. Ferritin has to be degraded, na? That's why whatever they store, it's like a snatcher showed you, whatever gun, took you somewhere. Now to nikalna hai na money. Nikalo. So how it degrades? It degrades because of proteases, acidic proteases present. There are about 20-23 acidic proteases in the lysosome. So which one is responsible? So we have done some scanning, again, because there are inhibitors, group inhibitors. So different group. I'm just showing one data. So what we found, the cathepsin B, this is the mature form and mature form. You can see that is also increased by dopamine. And when we knock down cathepsin B, the ferritin degradation is restored. So ferritin is now present in lysosome, but it cannot be degraded because that acid protease cathepsin B should also degrade. So you see in a cell how many components are there to make something happen. Okay? So now we have iron released. Iron released. Now where the iron is going? No, there is another something. Now we told you that ferritin is moving from cytosol to lysosome. Who will carrying out? Somebody has to take it, na? It cannot float and just go. So there is a protein discovered, it's called NCOA4. That came in Nature, and we are doing almost at the same time, but anyway, we are lost. They're a very big group. So what they found, that usually when there is a need, like iron deficiency, that NCO4 is stabilized, which is the coolie to take ferritin to the lysosome. Okay, the carrier. So they usually are not much, but when there is a need of iron, that means then they stabilize these coolies, and then they bind more to ferritin and takes. And here there is no iron deficiency. It's only dopamine. Okay. So actually there should be more, not because now ferritin is degraded, so there is more iron rather than iron deficiency. It's completely opposite. So we have done a similar experiment and we found NCO4 is increased. Okay. So it should not be, but still it is, should not be. Why? Because it usually known, so far literature is known only with the iron deficiency it increases. We have shown by neurotransmitter is also increases. And we recently found it is happening through a transcriptional mechanism. That we have not published yet. But if we knock down NCO4, then you see they are restored. Okay, so the coolie is not there, all other signals are there, everything is ready, even lysosome is ready, everybody, but coolie has to carry it out. The carrier is not there. You see another component. And obviously that pulling ear and the head is coming, that experiment, IP and immunoblotting, we showed that NCO4 can still bind to ferritin. So these are the different ways. Now the next one is where the iron is going. Okay? Again, so it is not coming back to cytosol, that we are not showing. It is actually going to the mitochondria. Okay? So mitochondria, now iron is more. You can see dose dependent, estimated. And if we block LAMP2A for lysosomal activity or HSC70 by siRNA and see the mitochondrial, iron is not so much now. So that means this pathway, what we discovered, is responsible for ferritin degradation and taking iron to the mitochondria. Now, if you think that if dopamine is not there, and usually I'll not show much, I have a lot of data, but I'm not showing much. If the iron is not, why iron goes to mitochondria? Two major things: iron sulfur cluster synthesis, which is important for TCA cycle and or ATP generation, and heme, which is also important for this. And heme also has an extra work. Heme degrades by hemoxygenase and to reach the bilirubin and biliverdin, which are major antioxidants. And it has been shown by Solomon Snyder, who got the Nobel Prize, shown that bilirubin, biliverdin is a very exceptional antioxidant in brain cells. So what we found, because I see I'm talking too much, so I'll not talk about these. So if we come here, so if we come here, because I'm just jumping the gun, so these are the important thing. And just one data I'll show you, that ferrochelatase is the component which makes heme. Okay, so protoporphyrin to heme synthesis is dependent on ferrochelatase, which is also increased by dopamine. So that means, so in between there are a lot of evidences are there, but I'm trying to make it short because people are yawning more. So what is happening, that dopamine is degrading ferritin to take iron into the mitochondria, and there is now more iron sulfur cluster and heme synthesis happen, and that heme is important for maintaining the antioxidant capacity of the cells. If it would have been not there, what it would happen? Mitochondria would not get iron, so there will be no more iron sulfur cluster and heme synthesis. Because of this, mitochondria would not function properly. So that's why we say if there is no dopamine, Parkinson's disease, now you can get the explanation that why mitochondrial dysfunction happens. Our job was to prove whether dopamine has a role in maintaining mitochondrial function. So at least one way we could prove, we showed the evidence, we found the evidence that dopamine can regulate mitochondrial activity. Okay, so these are still unpublished, and so this is the cartoon to show that dopamine degrades ferritin, brings mitochondrial iron, and this way they also stop iron release capacity, because they are releasing iron and this iron is released from the cell, the job will be not there. It has to go to mitochondria. So they block the iron release capacity of the cell. So I did not show much about that. And so finally, this is the way you can explain. So implication of dopamine-induced ferritin degradation in astrolia is maintaining mitochondrial function. And that's why ferritin is degraded, iron is going there, and there are many other things are there. Again, it's a short time to say this. So Ross generation and mitochondrial dysfunction in astrolia are important features in Parkinson's disease. So if mitochondria does not function properly, that also increase reactive oxygen species formation. Okay? So any question about it? No? Now the other job is done. Has to be done. You are bored already? Sure? So other job was that to show how iron can deposit in neuron. So we talked about mitochondrial dysfunction and iron accumulation. Okay? So one thing is that if astrocyte cannot degrade iron, iron will not go to neuron. Okay? So iron will be not there. So there is a very recent paper just published few months back. It showed that nigrostriatal iron accumulation in the progression of Parkinson's disease. This is a Nature Journal. Nature Journal exclusively published work on Parkinson's disease. So that means you have accumulated iron that will worsen your Parkinson's disease. So we must know that why this iron deposition is happening, what is the cellular mechanism. So here almost the similar mechanism, but here iron uptake is by transferrin receptor than DMT1. And there is another big player here. There was ceruloplasmin, if you do remember, but ceruloplasmin is not present in neuron. But there is a very big player called APP. So APP is amyloid precursor protein, which is linked with Alzheimer's disease. And what it does actually, so everybody of you say, oh, APP, I'll do research on APP because it's linked with Alzheimer. But Alzheimer comes when somebody is 65. So what, these 65 years, what they're doing? Most of the time we forget this, that what is the normal function of APP. So there are many actually, but two. I think let first show these and then I'll come back there. So what is the normal function of APP is that iron export activity is maintained by beta APP, and if it is not there, then that is cause Alzheimer's disease. So how it does, this is a Cell paper and this is a Nature Medicine paper. What it does, APP actually takes the ferroportin, which is the only unique iron exporter, okay, to the membrane. It is being synthesized in ER, but somebody has to carry them to the membrane to take iron out, okay. That is the important thing is APP doing. So APP has a role in taking there and stabilizing it. That is the Cell paper. But when we checked, we found APP is actually degraded. Down regulated, then we published that it is actually degraded. So APP is definitely not that player which is playing the role of stability. So our job is to also find out whether ferroportin is at all regulated and how it works. So we checked transferrin receptor iron uptake, no change. But when we checked ferroportin, which is the iron release component, now you can see this is being increased, okay, by Western blot, and this is in that iPSC derived neurons, pluripotent cells derived neurons from human brain, and you can see there is also increase of ferroportin, and this increase is happening due to increase in RNA level, okay, RNA level, and then I will not go very detailed. We identified a transcription factor called ANRF2, which is related to antioxidant capacity of the cell. So we cloned the promoter about 3 kb, and then promoter is cleaved and this etc., and found ANRF2, and then by point mutation, and this is cheap chromatin immunoprecipitation, we showed the ANRF2 is increasing the ferroportin transcription. Now, if you have to block ferroportin, if you knock down NRF2, the ferroportin will block, and you can play with them then with the biochemical function. So surprisingly, what we found when we knock down NRF2, you can see ferroportin is still being upregulated. We have done all those experiments at molecular level, it takes years to do, and then you say, oh no, if you knock down NRF2, still ferroportin is upregulated by dopamine, but the extent is less. So that means there must be another mechanism which is involved for ferroportin. And what is that? Then we guessed it must be ferroportin stability. Okay? So to prove that, what we did, we cloned the ferroportin with the flag, and you see that is increased. It is telling that this ferroportin is stabilized. And also ferroportin GFP, we tagged, transfected, and in a microscope you see there is now a lot more GFP. That means there is a lot more ferroportin. So dopamine is now connected for iron release. Say astrocyte, it was opposite. I didn't show that, I just mentioned their ferroportin is down by translation mechanism. They want to keep iron in astrocytes. But here it is more. From neuron, they want to release iron. Okay? So this I showed already. So we are searching in the whole literature what could be an alternate candidate for ferroportin stability. So there was one copper containing protein is found in gut, in intestine. It's called hephaestin. Okay? So it is again a cousin of ceruloplasmin. So this is a copper containing, multi-copper containing protein. So this is intestine, but nothing is known about the neuron. So what then we checked that what happens to hephaestin? If ferroportin has to be stabilized, hephaestin has to be increased. Okay? So we did Western blot in this is a hippocampal. So and then you say, oh no, if you knock down NRF2, still ferroportin is upregulated by dopamine, but the extent is less. So that means there must be another mechanism which is involved for ferroportin. And what is that? Then we guessed it must be ferroportin stability. Okay? So to prove that, what we did, we cloned the ferroportin with the flag, and you see that is increased. It is telling that this ferroportin is stabilized. And also ferroportin GFP, we tagged, transfected, and in a microscope you see there is now a lot more GFP. That means there is a lot more ferroportin. So dopamine is now connected for iron release. Say astrocyte, it was opposite. I didn't show that, I just mentioned their ferroportin is down by translation mechanism. They want to keep iron in astrocytes. But here it is more. From neuron, they want to release iron. Okay? So this I showed already. So we are searching in the whole literature what could be an alternate candidate for ferroportin stability. So there was one copper containing protein is found in gut, in intestine. It's called hephaestin. Okay? So it is again a cousin of ceruloplasmin. So this is a copper containing, multi-copper containing protein. So this is intestine, but nothing is known about the neuron. So what then we checked that what happens to hephaestin? If ferroportin has to be stabilized, hephaestin has to be increased. Okay? So we did Western blot in this is a hippocampal. So and both cases you find that hephaestin is increased. This is by Western blot, this is by immunocytochemistry, and this is in primary neuron. So not only ferroportin is increased, but hephaestin is also increased. And if you see co-localization, there is more yellow signal. Green and red, it becomes more yellow in presence of dopamine. So that means it is synthesizing more ferroportin, more hephaestin, and also they are co-localizing, so they can release the iron. So to prove it, we knock down hephaestin, and you see ferroportin flag, which is more stable, is now affected, both in the basal level and induced level. So you have to compare from these and these, these and this. There are already some, so you cannot take everything out. So both cases, so that means here ferroportin is stabilized. Now how it is doing? We are trained in chemistry. So what I say, it is peroxidase. This is a copper link with iron. Peroxidase means ferrous should be ferric by this enzyme. So this is if you take ferrous, and then highly ferric, if you can do biochemically and see with this ferrozine dye, the color is changed. So more lighter color means more oxidized of iron. So if you take neuronal cell, okay, and do the same experiment after dopamine, you say that means there is more oxidized iron than ferrous iron. And that's why iron. And if you take out the copper, because it's a copper containing protein, you see it just happens opposite way. Okay, I'm not going, hephaestin is not changed, but the activity is changed. That's the way copper is linked. And if you knock down hephaestin, you see the oxidation is affected. Any question about it? No. You are so much tired, then it's difficult to ask question now. So now to prove whether really dopamine is throwing out iron from the cells. So how do you prove the cellular iron level? It's very difficult to estimate that whether there was only 5 nanogram to 7 nanogram, 7 nanogram. So what we use sensitive techniques. So one technique is that FACS analysis. So whatever iron, we probe with some kind of iron fluorescence probe. Okay? So this is a control cell, you can see whatever, but if you add more iron, it moves to left side. FACS, you can see it's moved to more left side. So that means if cell has more iron, you will see left side. When there is iron chelator, less iron, it moves this side. So we have done same experiment with dopamine and see it moved the right side. That means there is lower iron, which is again shown by confocal microscopy. And what we found, so this probe, what it does, it is fluorescent. So if there is more iron, if you add this probe, the fluorescence will be quenched because it is binding with iron. If there is no iron, then it cannot quenched, so you can see more fluorescence. Exactly, because if there is no iron to bind with the probe, so that probe is showing more fluorescence, and this is done in primary neuron, that IPSC derived neuron. So now if we summarize that what is happened, that in healthy brain, dopamine is not affecting iron, but is regulating ferroportin both at mRNA and stability level, and that's why iron can be released. So neuron is balancing the iron mediated by dopamine. In case there is a Parkinson's disease, that means your dopamine is low. So still iron is coming in because it has no role in bringing iron. But that iron cannot get out. So that means in neuron, now there is more iron. Okay? That iron now can take part in ferroptotic mechanism to degrade the neurons. Okay? And then all this problem of Parkinson's may compound it. So this is I think we are almost done because you have 5 minutes or 10 minutes, whatever you can ask. So this work has been done by, astrocyte has been done by 3 persons, Somdev, who is now a faculty in AIIMS. Okay, Pratibha, she is now doing postdoc in US, and Rishmi is in London doing a similar work. That is the advantage of this field. You can go anywhere. Sanju is a head scientist in Biocon. Samya just recently last year, and Chainika is still working here. Although she is working more on that brain glioma, okay, cancer cell, but she helped doing some experiment. And with Professor Pankaj said in National Brain Research Center, we work on stem cell because we don't have that kind of all infrastructure to do iPSCs here, so that experiment we do there. Okay, and thank you very much. This is very nice. When iron is working for your way, you can defend it like a cannon. But somehow it is not working. Iron is oxidized, then rusting comes, and the enemies are coming. It will not work, or it changes the way it affects you. Okay. So this is on the top floor of Jaipur Fort. Okay. But I think this is the take-home message. Even you have to do iron in the pants or your saris or these. Otherwise, they will feel like anemia. Okay. You see, this is a trouser. What happens? Because it was not ironed.